Welcome, everyone. I'm Yamina from Dr. GPCR. I'm here today with Maria Mayalaro, CFO and co-founder of Saltaris Research, who will be co-moderating this webinar with me. Today's session is brought to you by Dr. GPCR and Saltaris Research. Saltaris Research develops fluorescent chemical tools and screening services for GPCR drug discovery, and they are the distributing team behind the probes you'll hear about today. Our speakers are Dr. Johannes Broichhagen, JB, who leads a research group at the Leibniz Research Institute for Molecular Pharmacology in Berlin. He develops chemical tools to study GPCR biology. Our other speaker is Dr. David Hodson, who is the Robert Turner Professor of Diabetic Medicine at the University of Oxford, where his group investigates GLP-1 and GIP receptor in complex tissues and environments. Many of you may already know them from the Dr. GPCR podcast, from episodes 168, 177 to 179, where these three speakers and our moderator, Maria Mayalaro, joined us together. Today, we take the conversation live. Before I hand it over, we have a few housekeeping items. Please feel free to type your question in the chat. If you'd like to ask questions live, please wait until the end of the webinar. Then we will call upon you. Please turn on your microphone and camera when you ask your question. David, JB, the floor is yours.
So thank you so much all for coming to this webinar, where we want to try to get to tell our hands-on experience on DAR Luxendins, dual agonist Luxendins for glucagon-like peptide 1 and GIP receptor labeling. So as Yamina mentioned, we are a lab of chemists and pharmacologists as well as biologists, and work at the interface to interrogate cell surface proteins. So I want to give a very brief introduction to the GLP-1R, or the glucagon-like peptide 1 receptor. It is responsible for the so-called incretin effect, and that is being mediated by this receptor. I hope you can also see my laser pointer. So when you eat and you take up glucose over time, the glucose will be lowered again, and this is due to the fact that this gets metabolized in the pancreas. When you already inject this, so when you eat it, you have a different response than when you actually put it into your veins. And responsible for this is the glucagon-like peptide 1 receptor. I know I'm preaching to the choir, but just like a couple of brief sentences. It is a class B G protein coupled receptor, and it is a drug target in the treatment of type 2 diabetes mellitus as well as obesity. What I found fascinating about it is that we know its cryo-EM structure, so we have atomic coordinates of where which amino acid and where which residue sits. And we know its canonical function, of course. It senses GLP-1, so the hormone that is released from the gut after a meal. It is involved in the insulin release from pancreatic beta cells, so it is also a marker for these cells. And it has become a blockbuster drug target with the sales a couple of years back exceeding nowadays the $14 billion mark. This is an islet of Langerhans, so the endocrine part of the pancreas. And as I mentioned before, GLP-1R sits in these beta cells, which are predominant in this mini organ. There's the insulin secreting cells. There are some alpha cells and delta cells, which secrete glucagon and somatostatin, respectively. And I don't want to go into the signaling details, but of course, it is a complex tissue setting, and a lot of different pathways are involved. So over here, you have the sugar that is entering the cell and then gets processed into ATP, which then closes potassium channels, ultimately leading to insulin secretion on the right-hand side. And then this is boosted by GLP-1R over here, which is a GS-coupled receptor, activating CAMP and putting out more insulin after in response to sugar inflow. And as I said, we're looking at not at a 2D cell culture mainly, but David will tell more about this in the complex tissue setting that is in the pancreas and in the brain. And we have other challenges here than when we just use immortalized cell lines. So, and furthermore, what we all know about this receptor, there are still some things that are hampered in our research. So we do not know about its fine interplay, that is its localization, its exact localization, its nano-localization, its dynamics. So how does it behave, laterally movement within the membrane, or what other proteins or other biomolecules does it engage? How many oligomers does it form, or does it form any? This still remains debated, and this is especially the case, as I mentioned, in the native tissue context. So the complex native tissue context has been shown here. So as you look at a cryo-sliced mouse embryo that was stained against the nuclei of the cells, so the DNA using DAPI, well-known small molecule, and then an antibody, actin in green, that stains the cytoskeleton. And then we used one of our Luxendin probes, Luxendin 645, in red in this case, and you can see it is distributed in different areas and in different organs. I want to be a little bit careful about this picture, just more like want to show it for aesthetic reasons at this point, as to date we haven't controlled this against knockout tissue, which is an important validation strategy. However, what you can see is the red staining over here on the lower left-hand side, the olfactory bulb, where GLP-1R has been reported, the salivary gland. Over here is the pancreas. Probably the dots are the islets of Langerhans. And then there are other organs where it does not seem to be expressed. So this is the heart, the lung, the liver, for instance. Here we have the spine and the rib cage. So the strategies for protein labeling, and when we look at this 7-transmembrane helix receptor, GLP-1R, with its active domain outside, traditionally people have fused fluorescent proteins to it, usually on the intracellular part, so on the C-terminus, while the N-terminus resides on the outside. Antibodies, of course, can be used that carry fluorophores, marked here as these bright shiny dots. You can use peptides or small molecules that engage the orthosteric side so that they bind to the active domain or partially to the inside of the 7-transmembrane helices. Or more recently, SNAP and HADO-tags, so self-labeling enzymes that are actually suicide enzymes reacting with a specific substrate, can be leveraged with fluorophores. So these are the general strategies, and they, of course, like everything in life, comes with advantages and disadvantages. So for instance, fluorescent proteins need to mature, so releasing reactive oxygen species, usually hydrogen peroxide. Antibodies are very difficult to use in the live tissue setting. SNAP and HADO-tags, as well as fluorescent proteins, need genetic engineering. So you would have to create a mouse, which, by the way, we also did. We're not going to talk about today. But like addressing this pharmacologically with peptides or small molecules is actually very attractive, since, as I said, they're usually tiny and can be applied in the live tissue setting without the need for genetic engineering. And of course, there's the perfect label, which we have not achieved to date. But characteristics it should bring, it should be bright, of course. So many photons you get out under the microscope. It should be stable, specific, and ideally also small. And these were the guidelines what we addressed the GLP-1 receptor with. That Luxendins has developed our peptidic antagonists, and then were designed in a way actually based on cryo-EM structures, where we see the transmembrane helices over here residing in the lipid bilayer, and the 8th helix over here is the C-terminus, which is not fully resolved, so it actually extends a little bit further. And then as a class B G protein coupled receptor, you see the active domain, which looks like on the extracellular side, spanning into the intercellular part. And in black, what you can see here is actually the peptide that is bound to this active domain with very high affinity, and also partly like buried into the 7-transmembrane domain, where it actually leads to activation of this G protein coupled receptor. So now when you want to fuse in a chemical way a dye or something to mark it to it, you know exactly which position you want to tackle. So we know the N-terminus is sitting over here, like in the receptor, and adding anything chemically to it would hamper probably binding. So what we decided is to go on the C-terminus, as this is not even fully resolved. So assuming, like stemming from cryo-EM structure, that this part is somewhat floppy and can be derivatized without any hampering any binding. So the idea is to put a dye here, and what we then did was we looked at an antagonist peptide extended 4, position 9 to 39, which is marked over here, and we did a tiny modification to the strong antagonist, and that was we replaced on the C-terminus this serine with a cysteine. So it's a minimal perturbation, changing essentially an oxygen atom to a sulfur atom, leaving the N-terminus untouched. So this can still like sit over here and towards the lipid bilayer. And with the cysteine, of course, we can do Malay-Mad conjugation chemistry. So now we can attach the dye highly selectively to this peptide. And then after HPLC purification, get different Luxendins out. And the great thing is we can just choose the color. So we can use a green color or like more yellow or orange or go to the red. And the nomenclature then stems from the maximal absorbance wavelength of the apparent Luxendin that would engage with GLP-1R. So my part of all of this is mostly the chemistry and the design and questions we often have. And that is something we want to address in this webinar is like how to handle Luxendins. So they are distributed via Saltaris Research, you can buy them. But then the vial arrives, and then the question is like what to do with it. And I think these are some not so trivial but important points to address also in this webinar. So one thing is the reconstitution. Usually we use distilled water or a mixture of DMSO water, and this 1:1, and dissolve a stock concentration to 100 micromolar. The Luxendins themselves are highly soluble in water. With our later version, and Dave will talk more about this, the dual agonist Luxendins, they need some DMSO for reconstitution. And this is already the one crucial parameter. So what people often use, like they have the big 1 liter bottle DMSO standing in the lab for a year. Maybe it has been exposed to the sun, it smells already a bit, so it has been oxidized or reduced, and it's not good anymore. It might actually destroy the Luxendin that arrived in your lab for your research. So what we recommend is DMSO ampules from DMSO D6, which is actually used in NMR spectroscopy. And they come in an ampule, which you can just like pop off the lid and just pipe it out, very fresh, very dry DMSO. And as they like also come in things, you can shield them from light. And they will never get exposed to air or anything they can react with, since the glass is fully sealed. So we get our from Roth Chemical, but you can get this also from other vendors as well. Another handling step is to pipette carefully up and down. Luxendins, we supply them when we did this back in the days when Saltaris was not involved from our research labs, and we lyophilized the samples directly from the HPLC, giving a powder, and sometimes there's a little bit of a spray. I think Saltaris is handling this differently, and like make sure that everything is on the bottom. But still, I would pipette it up and down and check for residues on the walls of the Eppendorf tube. This is not so difficult actually to do, because the Luxendins come as colored material, so you can spot it immediately if there's like something red or something blue on the side of the wall, which you also want to reconstitute. And then with this, you can dilute it in virtually any buffer at a concentration of 1 to 200. So you're getting a 500 nanomolar working concentration, which is actually still pretty high. So you can go down to even like 50 nanomolars and dilute 1 to 2000 if you want to work in more sophisticated or more sensitive tissue. But this works very well then when you have the stock concentration. What we also recommend is aliquoting to 1 nanomole. So usually you're supplied with a 3 nanomole vial. So the first nanomole you usually use straight away, and then the other 2 I would aliquot and store at -20 degrees. It's fine to just have it in the freezer. You don't have to shield it particularly from light, because usually in the freezer it's dark anyway. However, wrapping it in aluminum foil usually does not hurt. And if you want to lyophilize it again and get it a powder, we reported on a mixture of solvents called JB Special that we've published this year. It's acetonitrile, water, acetic acid. So all components are actually, I made this year, all components are amenable for cryo-freezing and then for lyophilizing. However, be careful with the acetonitrile. It is flammable, can be toxic, and keep it away, of course, from your eyes. I would also use it in a fume hood, and then keep it away from heat and ignition sources. Wear gloves, goggles, and a lab coat for this. So checking upon Luxendins. If you want to make sure like what you have in your vial, what do we recommend here? Is concentrations can be assessed by UV-Vis and Lambert-Burr law. So you have your stock concentration diluted further, like 100 micromolar are too high, and then make a dilution series, and then check via the extinction coefficient that is also provided. So for Luxendin 551, for instance, it's 150,000 per molar per centimeter. For Luxendin 645, it's 250,000 per molar per centimeter. And it's best to measure dilution series, which you can see over here, which you can then fit according to Lambert-Burr. So actually the slope will be the extinction coefficient, and then you can just like get your concentration. If you want to assess the physical binding constant against the GLP-1R ectodomain, we were very successful recently via surface plasma resonance. So you can actually buy the GLP-1R ectodomain from a vendor. It has a FC tag, so you can put it onto a chip. And then when you perfuse in the Luxendins, it gives you these responses in well, it gives you the responses with different concentrations that you actually do perfuse in, and then you can fit your KD bag. And the great thing about this is it doesn't matter if it's non-optical, so it doesn't matter like which color you actually have of the Luxendins. It works very well for whatever conjugation you've been using. So Luxendin specifically labeled GLP-1R, and this I just want to show you before I give over to David, is then when you apply these to the islet of Langerhans that I introduced before in normal wild-type tissue, which is GLP-1R positive, you can see the Luxendins stain the parts on the cell surface, while the nuclei are stained with GLP in blue. And also like testing this against an antibody. Of course, this is then in fixed tissue, gives quite good colocalization. And an important validation for checking this specificity is to use GLP-1R knockout mice and harvest the islets and there apply Luxendin and GLP-1R. And I hope you can appreciate and believe me that we think, or that I'm quite confident to say that we have a specific probe. All right, then I would hand over to David. And David, you give me a light when I should switch to the next slide.
Sounds good. So yeah, thank you from me for your attendance here. As JB said, we've been developing these ligands over many years now, initially obviously for ourselves, but it turned out that they were very useful because antibodies don't work very well for many GPCRs. So we did a lot of legwork to make sure that these would be really good. What I want to do is show you some examples of how we use them in the lab, which might also be informative to how you might want to use them in your tissues. So if you want to just ping the next slide, JB. So a lot of our work is in pancreatic islets, and what we use Luxendins for is to highlight specifically peak cells. We're going to show you just how specific that is later. And a lot of questions we used to get are that sort of as chemical probes, and because your islets are this kind of 500 micron spheroid, are they just going to kind of like label the GLP-1 receptor at the cell surface? And there was also a prevailing attitude that everything in the middle of this spheroid must be dead because of hypoxia. But it turns out that you can see from these two proton images here, you can pretty much label every cell within a spheroid type situation. And we see the same when we impact use tissue. So these are highly penetrating dyes as well. So that makes them quite suitable for lots of different systems. Next, JB. So this is an example here where we used a reporter approach. So these were mice which had an MTMG reporter. And what that does is it labels beta cells in green and all of the cells in red within the islet. And there was a lot of debate over where the GLP-1 receptor was at this time. Many people thought that the GLP-1 receptor was in alpha cells, and that explained the kind of suppressive effects of GLP-1 receptor agonists on glucose secretion, whereas others thought that that wasn't the case. It turns out it isn't the case, that alpha cells don't have the GLP-1 receptor. You can see that quite nicely here. You have the white stain, which is the Luxendin, and you'll see these big holes or gaps where we have the alpha cells labeled here in red. And what this means is, particularly if you work in islets like we do, the GLP-1 receptors become a really beta cell specific label, and it allows you to now sort those beta cells using the GLP-1 receptor as a surface marker for transcriptomics, for proteomics, for whatever it is you need to do. So that's coming out really useful for the field. Next. I forgot I animated that slide as well. Do you want to just next that one as well, JB? Because I think things are going to pop up. So when I animated that, I didn't realize that somebody else was going to be driving the slides for me, which is a learning point for me. What was really interesting for us actually was when these studies were done in 2020, there was virtually no information about how GLP-1 receptor agonists access the brain. So we knew they must act in the brain to suppress food intake and suppress appetite. But it wasn't really very clear how a kind of pretty, sort of charged peptide could get into brain regions beyond those that were freely accessible to the blood-brain barrier. So what you'll see here is animals that we injected IV with Luxendin. This was about 3 nanomoles. And then we sacrificed those animals about an hour post-injection. And you'll see that the Luxendin absolutely lights up the median eminence. So within the median eminence, you have sort of highly penetrated capillaries actually, which then allows substances to diffuse into the arcuate nucleus, and the arcuate nucleus is really key for the regulation of food intake. Very postrema as well, but also it can be involved in some of the side effect profile of the drugs, for example, with nausea. And we call it plexus, which we weren't expecting, but was actually really interesting because the colloid plexus generates your cerebrospinal fluid and your intracranial pressure. And it turned out that there was a disease where you produce too much cerebrospinal fluid called idiopathic intracranial hypertension. And it turns out that the GLP-1 receptor agonists were treating patients with this disease. And it was reducing CSF production from epithelial cells within the colloid plexus. So that was really intriguing. So intriguing, actually, that based upon some colleagues' work at Birmingham, NASA then took that forward because they have issues in spaceflight where astronauts can't manage their intracranial pressure because there's no gravity. So they get visual disturbances after about 6 months in space. So they were looking at potentially giving these astronauts symptomatics to help to manage these sort of disturbances that can occur. And sort of the skeptic signals tell us that maybe it would reduce food intake and increased payload required and kind of the spacecraft. But it was another case where sort of all this kind of identification of receptors really led on to, I think, some quite translationally relevant work. And again, that was read by Alexander Sinclair in Birmingham. It allowed us to go a little bit deeper as well, begin to look at receptor nanodomains. This was pretty early on, but I don't think we had any idea about where these receptors were in the brain or even how they were organized on the neuron. We'd spent a long time battling people showing receptors in the cytoplasm, which obviously just cannot be the case with 7TM. So it was quite gratifying to see this. Next. So me and JB obviously like to do imaging. So a lot of the time JB is developing these ligands so that we can push those boundaries, and that's kind of like our hobby. And then we use this information to dig out new biology. But with Luxendin 651, which is a silicon rhodamine conjugated dye, so that's fluorogenic, we're able to now do STED. So these were the first kind of endogenous snapshots of GLP-1 receptor at the about 60 nanometer level here. And this was in a beta cell plated up. And you'll see that they're not just kind of randomly aggregated, they tend to form what we now know as being a clustered nanodomain formation. And then we went one step further using sort of Storm, which gives us about a 30 nanometer resolution. This was with a Sci5 based probe, Luxendin 645, which is still really the best performing probe for most purposes. And because this is photoblinking, we're able to get these spectacular snapshots. And this was actually in an intact islet as well. And we went on to show that the nanodomain formation of these GLP-1 receptors is really critical for the signal profile. And there's lots of data coming out to support this as being actually therapeutically important as well. Next. And again, perfect. So we're colleagues over in the States in Amelia Miniman's lab. They do a lot of intravital imaging. And one of the issues is when you use mice, it's not always easy to use reporters, particularly in this case. This is a type 1 diabetes model called a nod mouse. They're very difficult to breed because they're sort of subfertile. They have large litters. They just a bit of a nightmare, really. So to then get these onto a background where you have a reporter, it was really tricky. So I thought, actually, we can use Luxendin as a reporter for the beta cells in this intravital imaging model. And this was interesting for 2 reasons. First of all, is these animals are kind of in the early to medium stages of beta cell destruction. So they have lowered insulin positivity, but they still have the GLP-1 receptor. And secondly, just how kind of bright the GLP-1 receptor staining is in vivo for intravital imaging approaches. And this could be the same for the brain or elsewhere that you want to look at. Next. So the Luxendins have been really useful. Both for us, we continue to use them in most of our kind of research programs and research questions. They're just really kind of easy to use. And they'd be really useful as well for a lot of other labs because there is, like I say, this issue with antibody staining for GPCRs, and it's well known in the GLP-1 receptor field. And it's a real minefield, and we're only just getting our head around maybe some of the reasons why this is. So I still think that these chemical probes are the best way to visualize the receptor, although, there are some caveats as well. So this needs to be done in live tissue and so forth. But based upon our experience and the experience of lots of colleagues, we then developed the dual agonists. So we call them dual agonist Luxendins or Luxendins. Dual agonists are very effective. So they are essentially a unique molecular design, which has components of glucagon, GLP-1, and GIP with stabilizing moieties, the C20 side chain to attach a lipid onto for circulating half-life. And this sort of sex spacer. And it's really quite an interesting story because two companies were making these drugs. Novo had the drug that 50/50 hit the GIP receptor and hit the GLP-1 receptor, but didn't show superiority to just the GLP-1 receptor on its own. So when you engage both receptors equally, same as just engage a GLP-1 receptor, and Lilly hits on this unimolecular agonist, which turned out that it had what we call this imbalance. So it hits the GIP receptor more than the GLP-1 receptor, and that gives it superior efficacy. And we're still trying to kind of piece together how that kind of works. And, don't even get me started on how GIP receptor agonism and antagonism sort of come into the argument as well. It's a really complex area of biology, but it's actually really interesting area of biology because it's the rare situation where the drugs are in patients and they're working very effectively. But as scientists, we've been afforded the luxury to go back and piece everything together. Should really be the other way around. You should really know how these things work before putting them in patients, but it is not the case yet. So lots of interesting things coming up on GPCRs, and I think that a lot of these things we learn with these class Bs is going to be applicable to other receptors as well. But we used a similar approach to make the Luxendins. Next, Jamie. And yeah, this is just sort of an image of that. It's this dual engagement, slight beta-arrestin bias as well, which is leading to this little bit of glucagon secretion, which doesn't happen with the GLP-1 receptor agonist on its own, and this kind of whacking great load of insulin secretion. And the glucagon secretion is increasing energy expenditure a little bit, which supports some of the weight loss effects as well. And, we're now, in my view, sort of at the kind of plateau of where we want the weight loss effects of these drugs to be. 20% by 70% placebo adjusted is sufficient for most patients, to be honest. And the new generation of drugs, the triple agonists, is really going to be reserved, I think, for those that are refractory to these drugs for whatever reason, or that need the highest levels and fastest rates of weight loss. So if you're suffering from fatty liver disease and other things like this. So tirzepatide is sort of best in class, as it is. And this was the one that we decided to modify. So JV again. It's similar principle to Luxendin, but now we're just kind of using a GPG linker to pull on the fluoroball. But the key thing with Luxendin is still the dual agonist versus the single agonist is they allow to sort of label a lot more cell types. And it just shows the kind of increasing complexity of how these drugs are operated within a complex tissue setting. So a GLP-1 receptor agonist on its own, so Luxendin, is really just labeling beta cells and nothing else, whereas the dual agonists are labeling all cells in the pancreas. And this is really key because you need all the different inputs from glucagon, somatostatin, and insulin to kind of have this properly regulated system, because the whole kind of islet modus operandi is regulated secretion. You don't want to just keep pushing insulin really hard because it's going to end badly. That's what sulfonylureas do. They're all drugs for type 2 diabetes, but they tend to push and lead to beta cell failure ultimately. So we kind of want this regulated secretion. I think this is what these drugs are doing. Next. That's the different hormone types. We also noticed that these dual agonists are not just kind of engaging receptors as a kind of summation type phenomena. They seem to be actively engaging heterodimerization of these receptors. So if you look at Luxendin here in the sort of first layer of panels, you kind of get this sparse but still nanodomain labeling of the GLP-1 receptor. And this is in primary islets. If we use a GIP receptor probe, it does the same. But now with tirzepatide, we're labeling more receptors than would be expected with either agonist alone. We don't really know what this means. I think we're kind of delving into it a little bit more. But it doesn't look as though you just have one receptor triggered and then another receptor triggered. I think that these receptors are coming into sort of proximity to pools of psychotherapy, GS coupling and so forth, which is really giving synergistic effects. And I think this is going to be really interesting biology going forward as well, which is again going to be applicable to the GPCR classes. Next. So kind of a interesting result, but slightly disappointing result at the same time, was that a lot of the thought in the field was maybe part of the reason dual agonists are working better is because they're targeting different sites in the brain, or they're penetrating regions of the brain more highly, and that would account for them having stronger effects upon food intake and a slightly different side effect profile, so better tolerated essentially. So we dosed animals with a dual agonist, two different colors, including one that had a lipid side shade on, and just a single agonist. So basically the dual agonists are in yellow and blue, and the single agonist representation is in purple here. And what you'll see is they just label exactly to the same extent and all the same brain regions. So you can't explain the superior efficacy of the dual agonists simply because they penetrate more deeply into the brain or label more sites within the arcuate nucleus. And I think this is really important because if we look at neurodegenerative disease, it's where some of these drugs are now beginning to be trialed with, I would say, kind of moderate to low success. You can't really explain that these drugs are hitting some of those deeper brain sites because they just don't get there. I mean, maybe it's not sensitive enough to visualize it, but it's likely that it's by indirect mechanisms such as changes in neuroinflammation or effects upon supportive cell types like oligodendrocytes and so forth. But I think this is kind of really intriguing. Next. What we do see, however, is that the dual agonists do label tiny sites, which we don't see with the single agonists. And these are a really kind of evolutionary conserved cell type that regulate a lot of processes within the hypothalamus, including food intake. We also see obviously the dual agonists engage kind of subsets of GIP receptor positive, GLP-1 receptor positive neurons, and it's probably that symmetry combination of inputs which is driving the stronger food intake. But I suspect as well that the glucagon and the slight increase in energy expenditure is going to be an important adjunct, and I think we see that anyway with the new type of dual agonists, which are just a glucagon receptor component with a GLP-1 receptor component, which have similar efficacy to tirzepatide. And again, that's intriguing because glucagon receptors are not expressed in the brain, so you can't explain that from brain actions alone. So again, it's all revealing very interesting biology, but that's just a snapshot of how we use these ligands. But other examples where these ligands have become pretty useful, people have been looking at GLP-1 receptor and chondrocytes, and a recent paper in Cell Metabolism showing anti-arthritis effects. People have been using these in immune cells because there's GLP-1 receptors in immune cells or a subset of immune cells. You name it, people will be using it for various things. All I will say that if you do use these ligands, the islet is like an exemplar organ, right? It has a nice specific expression of GLP-1 receptor. It's supported by single-cell RNA-seq. Everything maps up. GPCRs are GPCRs, right? You're labeling these things a one-on-one ratio. You can't get around that fact. It's not an amplified system, an amplification cause its own set of challenges of non-specificity as well. So it really is contingent on the end user to have the appropriate positive and negative controls in place. And we would obviously always recommend the GLP-1 receptor knockout animals. They don't have any protein that will bind to Luxendin, but also be careful because there are other animals that still have truncated protein that will bind but not signal. But a good example would be to use an autologous cell system with the receptor, sort of autologously expressed, or even just using siRNA. But that's continuing on you guys as top quality scientists to kind of go through the motions on that. But don't expect miracles, basically. What I will say though is it's not like when you buy an antibody where you know that you're not going to get any information, you're not going to get any help, it's not going to work. And you're probably going to have to microwave, pressure cook it, blow your whole lab up to get some semblance of staining in 2 experiments, and then we'll just pull it from the market. The reality is these are very well validated. We support all scientists really heavily because we are interested in the biology as well. And what you sort of say on the tin, it is what you're going to get. And I think that's the whole aim, is just to make sort of antibody vendors really kind of up the game a little bit as well, because just in the same way that we're responsible with our reagents, I do feel that should be the case with antibodies as well. So I think me and JB, if you want to move to the last slide, are kind of happy to take any questions, but we really do appreciate your time, and we're always happy to answer things over email or jump on meetings.
Okay. Well, maybe you can start us off with one question.
Yeah. I would be happy to do that. Okay, so just a question for JB. So the Luxendin, so the GLP-1 tracers are antagonists, and from your point of view, how important is that this distinction when working in native tissues, and does it ever become a limitation?
The question is if the antagonistic properties may become a limitation.
Yeah, I mean, yes, the distinction when you work in native tissues, for instance, using antagonists also, and yes, if it can be a limitation in some, setups.
Yes. So we strive to use antagonists before, but we started with antagonists because we did not know about the localization of any of this. So what we did not want to trigger was actually all the activation pathways and especially the internalization of GPCRs. This is what we started just to have a look at like where receptors are. And as David mentioned, we're very imaging heavy also in our laboratory. So the first idea was like to squeeze out a couple of nice microscopic images. And then we more and more moved towards the agonists because David also like very intrigued into the biology in the islet of Langerhans and in the brain. So we wanted to mimic actually the actions that drugs on the market exert on their targets. A limitation, we did not observe any disadvantages or differences in, like, let's say, access to certain areas if you use an agonist or an antagonist. That being said, of course, the agonist will be uptaken and possibly being downgraded at some point. Agonist might stay longer on the cell surface. But in terms of engagement or tissue penetration, this was working very well for all of our probes.
Thank you so much. And just to take a question we had from some people who were interested in the probes, these probes bind equally, I mean, not equally, but they bind both human and mouse receptors, right? Both Luxendin and Luxendin, right? So, and then...
That's correct, yes.
Okay. Okay. And did you test it also other kind of experiments? Because it's also something that they ask us, for instance, other applications like flow cytometry, for instance, with these probes.
Do you want to answer that, JB?
No, go ahead.
So yeah, first with the human biospecificity, obviously GLP-1 receptor agonists are pretty much equipotent mouse. Very few differences. You don't need to worry about that. And we frequently cross over mouse and human receptor systems, pharmacologically and like that. Tirzepatide, it's a little bit more tricky in the mouse, and I think that's sort of why it's a bit more problematic to study, because it's got different affinity or different efficacy and imbalance of the mouse GLP receptor and the mouse GLP receptor. So in the mouse, it tends to be much more GLP-1 receptor dominant than it is GLP receptor dominant, whereas the selectivity in humans is kind of switched the other way. One of the kind of neat things with Luxendin, and I don't think it came because we meant to do it, it was just sort of a property that was serendipitously there, is it actually shows more selectivity or a better selectivity for GLP receptor in mouse. So it's more like a mousey tirzepatide, if that makes sense, and potentially better model than tirzepatide itself. So that's pretty intriguing. But yeah, GLP receptor, always be a little bit cautious about, but otherwise the mouse and human is pretty potent anyway, and it's fine. Everybody uses tirzepatide in mice. Facts, yeah, I mean, we use this day in day out in facts. It's mega for facts, and it's super easy. We're using this a lot in stem cell islets at the moment, and we're using this as a method to sort cells and purify cells and so forth. And it's really where it comes into it. So lots of people are actually buying it to use in facts, and that's a really good way of doing things. You don't need up-close microscopy to use these kind of probes. In fact, that's just like, we like to use up-close microscopy because that's kind of, the hobby side of the lab, isn't it? But actually just day-to-day use, facts, brilliant.
And to add to that, you showed a couple of microscopy techniques, but swapping the fluorophore is quite helpful, because then you're amenable to like different techniques. So in addition to what we've showed, you can do 2-photon imaging. You can do TURF, so that to look only at like the receptors that are residing at the membrane, for instance. You mentioned the super resolution techniques like STED and STORM, which need different dyes due to their underlying chemistry.
You can even put gadolinium on it if you wanted.
We had a gadolinium probe, we never published it.
A mouse MRI scanner, which we do actually now here, but turns out it's quite expensive to use.
Yeah. Thank you so much. And yes, also another one question related to the use of these probes. So in your experience, the stability of these probes also in the conditions of the experiments, if you comment on that by the experiment that you performed.
I'm going to let JB go first, and then I'm going to give an anecdote.
Okay. So the stability of like, I want to start like when you receive the vial, because that is also an important question. So as a lyophilized powder, we're still using it like from batches like more older than I think a year, probably even 2 years ago. So it's really fairly stable. When you have it in a good solvent, like what I mentioned, the fresh DMSO and water, and you aliquot it out at -30 degrees, we also use it over months. So there, I do not see any issue that like the dye is falling off. This is usually a concern with this Malaymat chemistry, that you have a retro reaction, and then the Cy3 would only accumulate in mitochondria. So this is perfectly fine if you handle it correctly. And David, your story.
Yeah, I mean, look, I mean, I'm not a chemist, so we probably don't treat things as respectfully as we should in the lab. The Luxendins are really stable. I mean, even diluted, they're really stable. We've only had issues once where I think it was just an aliquoting issue. And that's where we should have probably just checked with UV pits, but that's we'd be in a lab. And we did check with UV pits. But when you get them from Celtaris, there wouldn't be any issues. And if there was any issues, then we would get it sorted out quickly. The GIP receptor probes which we're not supplying could do upon request are a bit less stable. So that just needs some more attention. The double agonist probes we're using now, again, they look to be really stable. So once aliquoted, even keeping in the fridge, they would be fine. But again, just do what you would do as a scientist, which is aliquot it out, and then single use or defrost, keep in fridge and keep using. If you're anything like us, whenever you get anything in a lab, because we don't have enough money to do research, we tend to hack things. So if the kid says we're going to do 384 wells for like a ELISA assay, we'll hack it to like double or triple if we can. So what I would recommend is if you can get a low volume for incubation, even 100 microliter in a 96 well plate is more than sufficient to incubate your tissue in, and you can always take that off again. Probably shouldn't be saying this in front of Celtaris, and then put it on another, kind of well. It's really quite stable. The anecdote is I had an ex-postdoc and now a sort of assistant professor in the lab, and she was labeling islets and tracking the islets, and we tracked those islets for 2 weeks, and we labeled Luxendin on day 1, and it was still there at day 14. So that tells you something about the half-life of the GLP-1 receptor, but also in the antagonist state anyway, but also just about how good these guys are. And you can just image them again and again, and they don't really seem to degrade that badly. So yeah, stable.
Yeah, great word, stable. Okay, so yeah, so as you mentioned, Yamina, do you want to comment?
Yes, and maybe I want to piggyback on what JP and David were talking about, and you as well, about the stability and the use. And this is a question for the three of you. When people start using these probes, what's the one mistake or what's the one question that comes up repeatedly, and how would you, what do you tell people to avoid that mistake? We talked about, JB, about resuspension, and you gave us advice about how to resuspend. Is there anything else that usually comes up with these probes?
I mean, from my point of view, the resuspension is a key, because they are generally soluble, but sometimes that when you take them from the fridge and you have to resolubilize them, you have to take care in this process, also because maybe you are using different buffers, and so it's very important to be sure about the solution that you have at the beginning before the protocol. Then, as David said, then they are stable during the experiment. So I think it is very important.
It depends how well your lab is, right? If your PI is loaded, then don't need to worry that much because you'll mess it up and then just order another one and nobody will know. But if your PI isn't loaded, then you probably just, go back to basics, right? Check everything, just, spin it down, make sure there's no excess static, use anti-static, and if you have it, not necessary, but we do just our procedure. And then maybe just put a little bit of buffer in a 10th or, a fifth just to start to dilute and just see if everything's progressing okay. The game changer for us is obviously being able to do the UV-based calculations as well, because I think that's going to be useful for lots of different things. But I can't teach a biologist how to, be good at the bench, right? I mean, we all mess stuff up. I'm sure if somebody mess something up really badly, then we would sort that out as well. But And I think the other mistake is just to moderate expectations, right? It's like, it's a biological tool. You're the one that's devising the biological question, and you're your own expert in that particular topic, particular tissue and disease. So it's up to you to come up with the sort of biological positive and negative controls that make sense in your particular assay. Again, we're happy to advise on that, send plasmid site and all the rest of it, but yeah.
Yeah, I agree with David that one question that's raised very often is what do we compare it against? Yeah, and David usually mentions like take some pancreatic tissue where GLP-1R is one in this case. But the important thing for me or for us, for me particularly as a chemical biologist, is like that you can build actually trust, right, in your probes and in your tools, that people can use it, it works straightforwardly, and there's also a supply that is ensured over a prolonged time. So this is why we also teamed up with Saltaris. And in particular for the Luxendins, I think we were amazed ourselves how well it worked, because when we first got it off the HPLC and sent it to Birmingham, where David then still, I think he called me the same night and said like I can image the whole islet. So it's like it.
Nobody would trust the data though, because I kept showing people the data, and one of our colleagues, Stefan, kept saying, "Well, it's just all stuck in the extracellular space." And I was like, "Well, what do you mean it's stuck in the extracellular space, right?" And then sort of there was gaps in it as well, and I was like, "Why is there gaps in it?" And it just took us a little while to put it all together, but yeah, for sure, until we had the kinetic data, it just looked a bit too good to be true, to be honest. And we're all suspicious about that as a scientist, because we're used to like disappointment, if I'm going to be honest with you.
And to some extent, he was right. No, it got stuck like extracellular space just on the receptor.
On the receptor, yeah. But like I say, we have a high expressing tissue. Don't expect miracles in low expressing tissue. If the transcriptomics doesn't map on, you've got a problem from the get-go, right? So in general, if single cell RNA is showing statistical receptor expression, you're good to go. If it's not, I don't see why you would have that discordance. And at the very minimum, use RNA scope or something like that. So yeah.
Okay, so I don't know, but yeah, we should close the session, right? Okay.
Yes. I will. Maybe some last words from you, Maria.
Yeah. So yeah, I mean, as David and JB mentioned, in Saltaris we are already distributing the Luxendin probes, and we will launch soon the Luxendin, so the lipidated version, the 554 plus and 6660 plus. So they will be available soon. And so you will see it on our webpage and website when they will be available, but we're already receiving some contacts. So so thank you so much, Yamina, for arranging this webinar, and David and JB, as always, for your support and contribution with your great science. And so, yes, and thank you all for participating and for the very interesting discussion. So I think we close the session now, right? Mm-hmm.
Yes, thank you so much, Maria. You said it so well, so I will not repeat myself. Thank you, everybody, for joining us. David, JB, and Maria, please stay. Martin, thank you for the fantastic questions. Everyone, thank you for joining us. And then we'll see you at the next webinar, hopefully in the upcoming weeks.